So welcome everyone, and first of all, thank you to the organizers for inviting me to contribute to this fantastic meeting and for putting together such an exciting program. So today I have the privilege of discussing with you the topic of bone marrow niche contribution to MDS pathogenesis. So this is also somewhat a challenge. And to be honest, today I'll probably be raising more questions than providing you with answers, but I will, however, take really this opportunity to share with you some of the ongoing work in my lab in which we are trying to continuously improve our modeling strategies in order to be able to actually functionally explore this topic in more details in the human setting. So these are my disclosures. So as you all know, the bone marrow is the main site of hematopoiesis in adults. And of course, besides hematopoietic stem cells and downstream progenitors, this microenvironment contains several populations of stromal cells that closely interact and modulate the behavior of hematopoietic stem cells to ensure the lifelong supply of mature blood cells. Now, because of their essential function in the past decade, tremendous efforts have actually really been put together to better understand the composition of the bone marrow niche, but also its potential relevance in the development and progression of hematological malignancies, including, of course, MDS. Now, this has obviously largely benefited from novel technologies, those being CyTOF or multispectral flow cytometry, single-cell RNA sequencing, that really further unraveled the complexity of these various cellular components that are within the bone marrow niche. Importantly, whenever the stroma or the hematopoietic cells are altered, this undoubtedly affects the multicellular crosstalk and can result in hematopoietic damage. And I will just, in the next slide, discuss a couple of studies that have looked into this in a bit more detail. So indeed, alterations in the bone marrow niche composition and molecular priming have now widely been reported, both in the context of physiological aging, but also in the context of infection and diseases such as myeloid malignancies. And a couple of years ago, the group of Nombela Arrieta actually used a population-based transcriptomic approach where they actually analyzed the temporal changes in the transcriptional landscapes with high resolution from juvenile, adult, and aged mice. Their finding actually shed light on the fundamental impact of aging on stromal cell biology, in particular, for instance, pointing to the acquisition of these inflammatory responses in the mesenchymal niche component. Now, besides aging, alterations in niche composition and priming are also seen in the context, or let's say in response to hematological malignancies. And here I'm just showing you a study from the Skadden group, also from a couple of years ago, where they actually could show these significant changes in the niche composition in response to MLL-AF9 driven AML. So with these mouse studies, these mouse studies are actually highly informative. However, they remain quite descriptive and do not necessarily address the major issue of causality. Also, similar single-cell studies in human are actually still missing, and hopefully a number of colleagues are working on addressing that at the moment, but it would be very informative to have such studies in the context of the human setting. There are, however, several clinical evidence suggesting that an aged and often inflamed bone marrow microenvironment may indeed provide a fertile soil where these genetically altered HSCs kind of strive and develop. In particular, HSCs that carry genetic alterations that are frequently seen in MDS and AML in the context of clonal hematopoiesis, or even MDS, are likely to benefit from such an environment. So this concept has now gained really additional evidence from a new study from a colleague of mine, Katharina Götze in Munich, within the CHOICE consortium, where they looked at a cohort of 200 patients that have been undergoing total hip arthroplasty, which is frequently indicated in osteoarthritis patients, a pathology that is driven by chronic low-grade inflammation. Now, the authors actually found that the prevalence of CHIP in this cohort of 200 patients was about 50% and these people were actually without known hematological disease, thereby really uncovering what was previously undocumented link between CHIP and autoimmune diseases. And interesting in this cohort, the mutations that were found in these patients are actually fairly the classical mutation found in the other in previous CHIP studied, such as DNMT3A, TET2, and A6N1. So even though this is really an HSC-centric study, it draws an important link, I think, between inflammation and CHIP. And however, I would say thus far, besides these clinical correlations, experimental studies addressing the contribution of an aged bone marrow microenvironment in human MDS or even pre-human MDS, let's say CHIPs, are actually still missing. And we believe that this is a fact that is largely hampered by the limited availability of experimental model. And this has long been the issue in the MDS field. And of course, in order to be able to address the critical question of the chicken and the egg, we need those models. So do we start with the genetically altered hematopoietic cells, or is it the stroma driving the development of MDS? So I think all of you must be very familiar with really what would always remain a landmark study in the MDS field that was put forward by Mark Keimaker when he was a postdoc in David Scadden's lab. And this study for the first time really showed that alterations in the environment can actually trigger the development of MDS. So in this study, Mark knocked out, specifically in osteoprogenitus, dicer, and observed the phenotypic development of MDS, and these mice developed pancytopenia. A few years later, we actually provided additional evidence in the context of a PDX model, in which we have shown actually that in order to propagate MDS stem cells into mice, we absolutely need to provide them with their mesenchymal niche component, thereby demonstrating that the stroma was actually essential for the fitness, or let's say the ability of these stem cells to propagate themselves in vivo. And this is just showing you basically that when we have high engraftment, we can actually see CD34, 38 positive cells that are exclusively carrying the genetic lesions that were found in the initial patients. Now, since then, I just want to highlight one study here from Dominique Bonnet's lab and just really point the people that in our study, we use a threshold for engraftment of about 1%. And if you use the same threshold in the Bonnet studies, you actually can really appreciate that when they transplant mononuclear cells with MSCs from MDS patients, they actually see a significant increase in engraftment, further confirming our results. So that was already a significant improvement. However, we wanted to basically go further, and an ideal model would actually allow us to reduce the number of required MDS cells. So this model is very informative, but tedious, requires a lot of CD34s from MDS patients. We wanted to avoid the irradiation and the intrafemoral injection, which is required in our modeling strategy. The main reason is that this is inducing damage to the niche. We wanted both from a postdoc and a PI perspective to reduce time and cost. And finally, our model did not recapitulate the aged environment. And I will talk to you later about actually also the idea that we are pursuing in the lab of having a fully human MDS system where we can have a modular and multicellular human environment to study those interactions. So in order to devise better model, we actually looked back into what are the most prominent age-related niche changes. So aging is accompanied by a prominent increase in adipocytes. So marrow adipose tissue is largely expanded in aged individuals. There's inflammation. The extracellular matrix is heavily changed, also as a consequence actually of the change in cellular composition. And we see very, very prominent changes in the vessel composition, as has been very nicely described by the groups of Sri Lapido, Ralph Adams, and Anjali Kosumbe. Now, our hypothesis, or let's say the approach that we have undertaken, is to try and understand whether simply by modifying the fate of the mesenchymal stem cell compartment, we can somewhat recapitulate many of those changes that are associated with the niche. And primarily, of course, this accumulation of adipogenic tissue. So besides collagen, the matricellular glycoprotein secreted protein acidic enriched in cysteine, which I will refer to in short SPARC, represent one of the most abundant proteins in the bone, and this expression is progressively downregulated in niche cells with age, both in mice and actually in human. And you can see it here in endothelial cells as well as in stromal cells. Now, Spark is known to actually inhibit adipogenesis and is upregulated during osteoblastic differentiation. We looked in human and sorted mesenchymal cells from young and elderly donors. And here we could also reproduce this strong downregulation of Spark in aged individual. Now we tested whether if we knock out Spark using CRISPR technology in a young MSC, in this case we had generated immortalized mesenchymal line, we can see that these cells, these clones, start adopting an adipogenic fate, really showing that simply by knocking out Spark, one could actually instruct those MSCs to change their fate and adopt an adipogenic cell fate. So we decided to use this strategy and actually generate a mouse model. The reason was also based on some data that was out there at the time, but also more recently. This is an analysis of mesenchymal cells sorted from MDS patients and explored in terms of their differentiation potential using CFU-derived colonies. And I think what you can appreciate is that MDS-derived MSCs have actually very little difference in terms of their osteogenic output. However, they show tremendous increase in their ability to give rise to adipogenic differentiation. This is shown here. And this is again a study by the group of Katharina Götze in Munich. So we decided to try and see whether adding a loss of Spark to our NSG modeling strategy would allow us to somewhat add another layer to our modeling, which is a phenotype of an age environment. So this mouse model was already described and generated by using CRISPR technology directly into NSG zygote. We generated several lines of Spark knockout here, just showing you that these mice indeed do not express the Spark protein, and within 3 to 4 months develop cataracts. So over the past couple of years, we've been actually looking into the phenotype of these mice, and of course, in particular, into their adipogenic phenotype. And I think you can appreciate here that if you look at the, this is whole mounts from femurs stained with pearly which marks adipocytes, you can see these very prominent, very rich adipocytes in the Spark knockout mice. What is quite remarkable is that these mice, even though they have a very high marrow adipose tissue, they do not show any gain weight compared to their wild-type counterpart. And this is very different from experimental models in which high-fat diet, for instance, have been used to induce increase in adipocytes in the bone marrow. Now, in addition, we see a very clear bone phenotype in these mice. The shift between the, let's say, the imbalanced output towards adipocytes also depletes the output in osteoprogenitors. There is an imbalanced osteoclast towards osteoblast that leads into reduced trabecular numbers and reduction in bone volume, all of which are pretty much reminiscent of physiological aging. Now, in addition to controlling adipogenesis, Spark has also been shown to be extremely important in stabilizing fibrillar collagen. And we know that the bone is 90% composed of collagen. So we looked into collagen, and we had some help from Diana Passaro at the Institut Cochin. Using 2-photon microscopy, we could see a very strong reduction in collagen 1, shown by second harmonic generation. In addition, we did immunofluorescence staining here, looking at collagen 4, and I think you can really appreciate that these Spark knockout mice have a tremendous decrease in collagen 4 expression. Spark protein, of course, is gone. And I think you can actually also see these vascular phenotypes with higher branching point, shorter vessels. And we documented also the lumen of these vessels to be much larger, and this is shown here. We see increase in median diameter and median volume in these vessels. And so taken together, we've been able to generate an NSG model in which mice exhibit a bone marrow phenotype that is reminiscent of physiological aging. And we believe this model will be very useful to actually study the interaction of hematopoietic cells from aging individual in an environment that is more closely related to the, let's say, the source of these samples. Now, one additional caveat we had to overcome is the need for irradiation and intrafemoral injection. And to do so, we actually decided to cross our Spark NSG mice with the KIT W41. Unfortunately, these mice were not available to us, so we again went back to our CRISPR technology into NSG zygote and generated the NSG KIT W41 mice, which allow you to actually reconstitute the mouse without the need of irradiation. And we generated these compound mice that we will use in the further studies. Now, to avoid the intrafemoral injection, we actually used an alternative strategy that was described by colleagues from Japan in a recent Nature Communication paper, which is the intracaudal artery injection, and basically transplanted now adult CD34 positive cells. And here I would like just to pause for a second and really make the remark that in many studies out there, if not all, usually people use cord blood CD34s, as it is extremely challenging to engraft and propagate adult, and the older they are, the more difficult it is in NSG mice. So these were actually donors that were around 40 to 50 years old, and we transplanted them into KIT versus the SPARC KIT mice. And I think you could appreciate here that the SPARC deficiency tremendously improved actually the engraftment of those cells. This is after 3 months, we see 60 to 80%, which we have never been able to achieve with adult and older stem cells. So this is really the system that at the moment is the most attractive for us in the lab, and we're more than happy to share this with any of you that would be interested to explore it in your studies. Now, we also wanted to develop systems that would be more amenable to, let's say, experimental manipulation ex vivo. And for this, we developed what we call human organotypic marrow environment in a 2D or a 3D format. The short for that, it stands for 2D or 3D homes. These are modular systems in which we can combine mesenchymal cells, endothelial cells, and MDS stem cells or AML stem cells, hematopoietic stem cells, in a co-culture setting in vitro, and this can be done in a 2D format or a 3D format by using a collagen scaffold. These systems are very highly amenable to ex vivo experimentation. You can genetically manipulate the cells, you can perform imaging, drag streams. They're fully human, in particular in the 2D setting and 3D setting if you maintain them ex vivo. They can be personalized because we can definitely isolate MSCs and ECs from patient bone marrow. They are definitely time and cost effective, and they require actually a very limited number of MDS cells. And I will show you some data on how we use these systems. So one of the challenges really to develop such a system was to be able to isolate endothelial cells from bone marrow biopsies. Most of the people use HD-MEC or HUVEC. So we wanted really to have the bone marrow derived ECs, especially given all the recent single cell studies showing a tremendous heterogeneity in ECs depending on the tissue source. So we devised a protocol to isolate these highly purified endothelial cells from any bone marrow biopsy, and also very much happy to share those protocols. We also managed to immortalize these endothelial cells from patients as well as from healthy donors. This is their growth curves. After passage 20, you can keep growing them in culture without any problem. And these immortalized ECs actually show a very bona fide phenotype that is comparable to the parental ECs in terms of VE cadherin expression, CD31, CD105, also shown here, von Willebrand factor by imaging. We also actually sequenced those immortalized and compared them to their parental, and what you can see, and compared them also to existing signatures of endothelial progenitor cells, either the ETV signature put forward by Shaheen Rafi or another study that was published in 2016. And I think you can appreciate that these gene signatures are actually highly enriched in our immortalized endothelial cells. And these cells are not transformed, they do not form tumors when they're injected into mice. Now, the 2D home system is very simple. You combine these MSCs and endothelial cells, and in this case, in the following slides, I'll be using the MDSL that are marked with GFP as a source of MDS cells. So at least in our hands, this particular MDSL subline does not engraft in mice if we transplant them into NSG mice, even though they are irradiated. So when we co-culture these cells together with this network of vessel-like structures, which nicely recapitulate the vascular niche in the bone marrow, we can see that these cells actually enter quiescence. And this has been already shown in the context of solid tumor that disseminate to the bone marrow. They actually enter quiescence, which is imposed by the vascular niche. And we use this system to actually test whether changes in the mesenchymal niche component were actually instructively induced by exposure to MDS cells, something that we had actually shown in a much more simplistic system during in our cell stem cell paper. Now, when we sort MSCs that have been cultured with MDSL or without, we can actually see enrichment of processes that are associated with TNF-alpha signaling, inflammation in general, TGF-beta signaling, but also induction of processes associated with adipogenesis, suggesting that part of these phenotypes that we see in MDS associated with MSCs are actually instructively induced by the MDS cells. What's quite interesting is when we looked at now samples that are directly isolated from patients. Here we analyzed peripheral blood from a cohort of young versus aged individuals, so peripheral blood plasma, we see this prominent signature of inflammation. And here we sequenced MSCs from young versus old donor. We again see enrichment in those particular gene sets that are actually instructively induced in our 2D home setting, suggesting that that system can really be used to interrogate the crosstalk and perform functional experiments. Now, another model that we have, or let's say another question that we have addressed with this model is the question of vascular alterations in myeloid malignancies. These have been widely reported, including in MDS, more, let's say, experimentally studied in the context of AML using mouse models. And it is actually in general believed that these vascular alterations are driven by the leukemia cells. However, we actually were wondering whether some of those alterations might be due to an indirect effect coming from the mesenchymal niche component, which is altered in those patients. And the reason we suspected that is when we look at the gene expression in MDS-associated mesenchymal cells. I think you can appreciate here in this qPCR data, there is an overexpression of a number of genes associated with angiogenesis. And so what we did is a very simple experiment. We took advantage of our 2D model and we combined, mixed and matched the healthy MSCs with healthy endothelial cells, healthy MSCs with MDS endothelial cells, and so on. And I think you can appreciate that as soon as you put healthy endothelial cells in presence of MDS MSCs, which is actually shown here, you see this reduction in vessel formation. And actually, this is a kind of a crescendo, not a crescendo, but a progressive decrease in the ability of forming proper vessels that is actually the worst in the combination between MSCs from MDS and endothelial cells from MDS. So really suggesting that part of maybe these aberration in vessel formation and endothelial cell growth versus differentiation might be imposed by direct interaction with the MDS MSCs rather than the MDS leukemic cells, or at least a combination of both. And this is just, again, an example of how the system can be used to address some mechanistic questions. And now I'll just move to the 3D format of such a culture. So in this setting, we combine MSCs and endothelial cells in a collagen scaffold. This is how it looks at day 1. We allow these cells to grow for about 7 days, and we see this gel contraction. At this point, we add the MDS cells, and we can implant them into a mouse before explanting it at 5 weeks post-transplant. And this is how it looks. You can see these bony structures being formed. You see some fibroblastic tissue as well as fat, and you see accumulation of MDS cells in there. Now, let me just remind you that those MDSL are not able to grow when we inject them intracaudally into NSG mice that have been irradiated. So they really need this human environment in order to expand. And what was also remarkable here is that we used endothelial cells that were actually marked with the tomato, so we can distinguish the human from the mouse endothelial cells using endomucin. The MDS cells are marked with GFP, and vimentin is marking the stromal compartment. And I think you can appreciate that now we have human cells growing in an environment where we have human stroma, both vessel, human, that are connected with the mouse vasculature. And I think this is as close as we get to a human patient. Now, what is the next generation, let's say 3D homes? We are actually exploring the possibility of adding immune cells. And that would allow us really to have a complete system in which we have at least the essential component of the bone marrow niche. We can, of course, also alter the ECM by using different scaffolding systems. And we hope that we can use this system for dissecting molecular interactions in the context of all of those challenging diseases such as MDS, but also possibly clonal hematopoiesis, which is of interest to our lab as well. And with this, I would like to stop and thank you for your attention. I'm happy to take any questions, but most importantly, I would like to acknowledge the people that did the work. So this work has been driven by Alexander Schaefer, Arnaud, and Irene in my lab. Our clinical collaborators, without whom this work would not be possible, Katharina Goetze from Munich, Uwe Platzbecker and Klaus Metzeler from Leipzig, Halvard Bönig from Frankfurt, the Bone Lab in Dresden for helping us with the bone phenotype, Aline Bozik for her adipocyte expertise, Mohamed Mosa helped us with the RNA-seq analysis, and of course Diana and Alaria for helping us with the 2-photon microscopy. Thank you.